Hello, and welcome to my presentation. My name is Antoinette Adams. I currently attend Grand Canyon University, and I am in the class Biochemistry. Today we'll be discussing signal transduction. Today I want to dwell into the intriguing area of GPCR signaling, which is just the gene protein coupled receptor signaling. These receptors play a vital role in our body's communication system, regulating a variety of physiological functions. Imagine your body as a busy city with many departments. Um, each department is in charge of a certain purpose. So I gave you this, analogy so you can kind of get the fact that GPCRs represent the city's communication network. Here, I'm sorry. Here you see where you have the receptor. It goes through this communication system, which would then have a cellular response. So once again, each department is in charge of a certain purpose. The GPCRs represent the city's communication network, which transmit messages across departments to guarantee seamless coordination and efficient operation. So why are they important? For their presence, one, GPCRs are only found in eukaryotes and is one of the largest and most diverse families of cell surface receptors found in nearly all cell types. This widespread presence underscores their significance in regulating diverse processes. GPCRs are also important due to that they are signal transductions. So when a signaling molecule or ligand binds to a GPCR on the cell surface, it triggers a series of events known as the signal transduction. This process involves the activation of G proteins, which acts as molecule switches, initiating cascades of intracellular signaling pathways. They're also good for their versatility. GPCR signaling is incredibly versatile. It regulates processes such as like the sensory perception, like vision and taste, neurotransmissions, your hormone secretion, immune response modulation, and even cell growth and differentiation. So the GPCR is very, very important. The GPCR is like it helps with cellular activities that keeps our bodies functioning. So throughout this presentation, we'll explore the mechanisms of GPCR signaling in more detail, highlighting its importance in both health and disease. So as previously stated, GCPRs is one of the largest class of membrane proteins. More than 700 GPCRs are identified. The most important is that they have 7 transmembrane alpha helices, which are sometimes called the 7 membrane receptors. Here you will find a conserved structure for the 7 membranes. Whenever you have an extracellular and an intercellular, if you have a G protein receptor, it will span around the plasma membrane 7 times. And so here's what you kind of see here with the G protein spanning around the plasma membrane 7 times. G proteins are specialized proteins that have the ability to bind GTP and GDP, able to bind both GTP and GDP. When a receptor is activated by adrenaline or a photon of light, it changes conformation, becomes activated, stimulates G proteins, leading to the activation of second messenger enzymes. Those second messenger enzymes activates kinesis, which leads to cellular response. So now here I have a photo of the three subunits of the G protein. So the heterotrimetric G proteins are signaling proteins composed of three subunits. You have the alpha, you have beta, and gamma subunits. They are involved in transmitting signals from activated G protein coupled receptors at the cell membrane to intracellular effectors such as enzymes or ion channels. Alpha and gamma are both attached to the cell membrane by lipid anchors. G proteins bind GTP or GDP. Inactive protein binds GDP, glutamate diphosphate. G protein receptors react to outer molecules in the outer cell. G protein coupled receptor binds to molecules fitting together like a lock and key. So keep in mind, G proteins bind GTP or GDP. So I'll play this animation here. Okay. So keep in mind that when the ligament binds, the G protein exchanges GTP in place of GDP and gets activated. Inactive protein binds GDP, which is the diphosphate. The GTP bound alpha subunit of the G protein, now active, interacts with and activates downstream effector proteins within the cell. In the case of adrenalic receptors, the G protein may activate an enzyme that produces CAMP or ciliate um, ciliate chondroitin monophosphate. So CAMP acts as a second messenger, triggering a cascade of intracellular signaling events that ultimately lead to cellular responses such as increased heart rate or glucose release in response to adrenaline. So here I'm showing the G protein interaction domain. You have the desensitization, the internalization, ureation binding. So you have the ligand binding domain, which is just a receptor. It's the part of the receptor protein that interacts with signaling molecules or ligands from the extracellular environment. This domain is crucial for initiating cellular responses upon ligand binding. So in GPCRs, the ligand binding domain is typically located on the extracellular side of the cell membrane. It consists of specific amino acid residues that form a binding pocket or site for lesions to attach. When a lesion binds to the lesion binding domain, it induces conformal changes in the receptor, leading to the activation of downstream signaling pathways through G protein coupled or other mechanisms. You also have the protein interaction domain, which is simply just a region of the receptor protein that interacts with the alpha, beta, and gamma proteins, which are signaling proteins involved in transmitting signals from activated receptors to intracellular effectors. In GPCRs, the G protein interaction domain is typically located on the intracellular side of the cell membrane. It contains amino acid sequences that interact with specific subunits of G proteins, such as the alpha subunit. Upon ligand binding and receptor activation, the G protein interaction domain facilitates the coupling of the receptor with G proteins, leading to the activation of downstream signaling cascade and cellular responses. Okay, you also have defossilization sites, which is like where you have desensitization, internalization. So sites are specific amino acid residues such as serine and tyrosine within a receptor protein that can be phosphorylated by protein kinases. Okay, so I'm going to pause right there and keep going. External signals are converted to internal responses. So the definition of a signal transduction is just the ability of the cell to translate receptor-ligand interaction into a change in behavior or gene expression. Cells sense and respond to the environment. Cells communicate with each other through direct contact or chemical signals. The process begins when a signaling molecule or ligand, such as adrenaline, binds to the adrenalic receptor on the cell membrane. Adrenaline is a neurotransmitter and hormone that activates the fight or flight response. Upon binding, the ligand induces a conformational change in the adrenalic receptor, leading to its activation. So I've been away at the bottom down here, but it leads to the gene expression. Next, we have the signal amplification. One of the key receptors relay signals via intracellular signaling cascades. Each protein in a signaling pathway amplifies the signal by activating multiple copies of the next component in the pathway. One of the key features of GPCR signaling is signal amplification. Once it's activated, a single ligand receptor interaction can lead to the activation of multiple G proteins, each of which can activate multiple effector molecules. This amplification process ensures that a relatively small number of ligand molecules can produce a significant cellular response, making GPCR signaling highly effective and sensitive to extracellular signals. GPCR signaling involves ligand binding to the receptor, activation of a G protein complex, activation of effector molecules, and signal amplification leading to cellular responses. The example of the adrenaline receptor illustrates how these steps occur in response to adrenaline binding, ultimately regulating processes such as heart rate and metabolism. So it's important to know that one primary signal activates an enzyme activity processes 100 substrates per second. Primary enzyme activates 100 target enzymes. Each of the 100 enzymes activates an additional 100 downstream target enzymes. Each of those 10,000 downstream targets activates 100 control factors. So you rapidly have 1,000,000 active control factors. Like, I find it very fascinating of how it duplicates and multiplies. Next, we will talk about the electrical signals. Electric signals. So electric signals fly through your brain. Here you have the action potential ion channel. So you have a nerve impulse, which is rapid, very fast. And so electrical signals fly through your brain and body, carrying messages to receptors and cells like very fast within a blink of an eye. Nerve impulses involve the opening and closing of ion channels. These are water-filled selectively molecule tunnels that pass through the cell membrane and allows electrically charged atoms or tiny molecules to enter and exit the cell. The movement of ions generates an electrical current, which causes small voltage changes across the neuron's cell membrane. Signal transmitted to muscle cell across the synapse. Here in A, The first part of the diagram demonstrates depolarization, which opens the voltage gate of calcium ion channels. Here in B, you see that the calcium ion channels rushes in, the vesicles fuse with membrane, and then at the end, at the bottom here, you have the neurotransmitter is released. It opens the ligand gated ion channels on muscle cells, which depolarizes the entire muscle cell. It's raining very hard outside. Okay, now you have the effector proteins and second messengers. An activated G protein can trigger the formation of the cyclic cages, the cyclic adenosine monophosphate. I keep trying to pronounce the entire name for CAMP. Which then acts as a second messenger in cellular pathways. Effector enzyme produces the second messenger CAMP. Second messengers such as cyclic monophosphate are small molecules that relay and amplify intracellular signals initiated by extracellular ligands, such as like your hormones or neurotransmitters, which binds to the cell surface receptor. These molecules play an important role in signal transduction pathways, far as like how they convert extracellular signals into cellular responses. And so it kind of all plays back into like, just kind of sum this up, this kind of all plays back into signal amplification, because like you have the activation of cell surface receptors, which is like the process begins when a ligand binds to a cell surface receptor. Like so the ligand receptor interaction triggers changes in a receptor leading to its activation. You also have the activation of downstream signaling, once again, which just activates the cell surface receptor and allows it to initiate intracellular signaling, through different ways. So, for example, the GPCRs activate, the three proteins alpha, beta, gamma, which leads to the activation of downstream effectors, such as philosophies. I messed that word up, but receptor tyrosine kinesis, on the other hand, activate intracellular signaling pathways involving proteins like RAS, which leads to the activation of downstream effectors like nitrogen activated proteins or MAP. It also generates the activation of downstream effectors can lead to GTP and GDP. The guanosine triphosphate and the guanosine diphosphate. So, for example, activation of the adrenaline by the G protein signaling results in the conversion of ATP to CAMP, which serves as a second messenger in many signaling pathways. This amplification process allows a signal ligand receptor interaction to generate a robust and widespread cellular response. So the activation of PKA by CAMP can phosphorylate and activate numerous target proteins, which can also lead to gene expression, metabolism, ion channel activity, and other cellular functions. Now, the G-coupled receptor signaling involves a receptor that binds a ligand, which then activates the entire process. Alright, so in turn, it activates PLC. This activation leads to the cleavage of a molecule called phyloselamine. Yeah. So IP3 is the short name. It plays a role in releasing calcium ions from intracellular stores, specifically the endoplasmic reticulum, by activating the IP3 receptor. So on the other hand, DAG is necessary for activating a protein called protein kinesis, which is PKC. You have the desensitization of GPCR signaling, which refers to regulatory mechanism that reduces the responsiveness of G protein coupled receptors to continuous stimulation by ligands. This process helps prevent excessive or prolonged cellular responses, maintaining cellular homeostasis. Here's how desensitization typically occurs. So it usually occurs by kinesis, which is just when the ligand binds and the receptor activates. The proteins such as GRKs and second messengers depend on kinases, like protein kinases A and protein kinases C. Phosphorylation occurs primarily on specific serine residues located on the intracellular loops and C-terminal tail of the receptor. You also have the arrestin binding, which just activates the receptor promoting the binding of the proteins to the phosphorylated sites. So arrestins are regulatory proteins that interact with the GPCRs, which leads to receptor desensitization and internalization. You have the binding of arrestin to the phosphorylated receptor prevents further interaction with G proteins, thereby halting downstream signaling pathways. You have the endocytosis and receptor internalization. So after the arrestin binding, the phosphorylated and desensitized receptor undergoes endocytosis, a process within itself. So according to a research that I found by Violin and others, it shows that the dysregulation of the GPCR signaling can contribute to various diseases by disrupting normal cellular responses and signaling pathways. In allergic reactions, the dysregulation of GPCR signaling can lead to an exaggerated immune response to harmless substances or allergens, such as pollen, dust mites, or certain foods. If you're like me, pollen is not my friend at all. Me and pollen do not get along, and I'm always having blockage and mucus and a lot of things. Another thing to look out for when it comes to the health of humans is heart disease. So the dysregulation of GPCR signaling can contribute to cardiovascular diseases such as hypertension, heart failure, and adrenergic receptors that regulate the heart rate and the vascular tone. So dysregulated signaling such as excessive activation of B1ARs can lead to increased cardiac workload, hypertrophy, and impaired cardiac function, contributing to heart failure and arrhythmias. It also, last but not least, can also lead to psychiatric disorders. So the GPCR dysregulation is, it's involved in various, I'm sorry, I'm having a brain freeze, and various, psychological disorders, including depression, anxiety disorders, schizophrenia, and bipolar disorder. Serotonin receptors are GPCRs involved in mood regulation and neurotransmission. Dysregulated serotonin receptor signaling, including altered receptor expression or function, can contribute to mood disorders, like depression and anxiety. I hope you had gained valuable information from our presentation today. As we went over and discussed GPCR signaling, how the alpha, beta, and the gamma all work as one unit, as a communication signal system, which sends the signal of the cellular response, which sometimes undergoes processes of desensitization, which will turn it down. Thank you.